Hello dear student, this lecture is about the carbohydrate metabolism, which is the second session of discipline biochemistry course. As you see in the title of this slide, instead of carbohydrate metabolism, I have mentioned glucose metabolism because glucose is the most important carbohydrate. In this schematic picture, you see the major pathway and end products related to the glucose metabolism. First of all, I should mention that these major metabolic pathway related to the glucose are divided to two groups. Okay, so the metabolic pathway that result in decreasing blood glucose level, and the pathway that result in increasing blood glucose level. Okay, those that are involved in this in decreasing the blood glucose level, they could be subdivided as well to the oxidative pathway and storage pathway. Okay, because the pathway that lead to decreasing blood glucose level, either this pathway oxidize glucose or they convert glucose to the polymer such as glycogen or to the lipid that could be stored in the cells of our body. So related to the pathway that decrease the blood glucose level, glycolysis is in this group, pentose phosphate pathway and oronic acid pathway. Okay, these three pathways that I have mentioned, they are in the group of decreasing blood glucose level and in the subgroup of oxidative pathway. In addition, glycogenesis and lipogenesis, these two pathways are in the group of again decreasing blood glucose level, but in the subgroup of storage pathway, meaning that they convert glucose to the glycogen and lipid, 3-acetylglycerol, which could be stored in the cells of our body. So, in addition to pathway that decrease blood glucose level, I told you that we have pathway which increase the blood glucose level. So the pathway including gluconeogenesis and glycogenolysis are among the pathway that increase the blood glucose level. As you see, there are arrows that leave the glucose. I mean, they go far from the glucose, meaning that they use the glucose and they produce these end products. But these arrows that coming close to the glucose, it means that they are producing glucose from, for example, here for the gluconeogenesis from the non-carbohydrate substrate. Okay, so as you got an overview from this pathway, I'm going to give more information about this pathway to you guys. So regarding the glycolysis, is the main pathway of the glucose metabolism, which occurs in the cytosol of all the cells, and it can function either aerobically or anaerobically. So therefore the product here depends on the situation of the cells, whether the oxygen is available or not. Okay, so in general, the glucose in the glycolysis pathway convert to the pyruvate and ATP if the oxygen is available and the mitochondria is available. The pyruvate is converted to the acetyl-CoA. Acetyl-CoA enter to the Krebs cycle in order to release its energy. But if the condition is anaerobic, so the pyruvate is reduced to the lactate and no more acetyl-CoA is present. So the pentose phosphate pathway is the pathway which convert the glucose to the ribose and NADPH. As you see, there is no ATP formed in this pathway, whereas the ATP is formed in the glycolysis. So the oronic acid pathway convert glucose to glucuronate, and the glycogenesis convert glucose to glycogen. I mean, it convert the glucose to a polymer of the glycogen, which could be stored in the liver and in the muscle. So in the pathway of the lipogenesis, the glucose is converted to the triacylglycerol, and triacylglycerol is the storage form of the lipid in our body. And as you saw, the glucose could be converted to the lipid in our body, but the vice versa is not allowed, meaning that the lipid could not be converted to the glucose. So, in addition to these metabolic pathways that lead to decrease in blood glucose level, we have gluconeogenesis, which convert the non-carbohydrate substrate to the glucose, and we have glycogenolysis that convert or break down the glycogen to produce glucose. These two pathways increase the blood glucose level. Okay, so in this lecture, I would like to talk about the digestion and absorption of carbohydrate. We will talk about the glycolysis pathway, the pyruvate which is produced by the glycolysis, how it could be oxidized. We talk about the Krebs cycle, gluconeogenesis, a pathway which leads to the production of the glucose. Glycogenesis, as I mentioned in the previous slide, this pathway produces glycogen. Glycogenolysis, the metabolic pathway which leads to formation of the glucose from glycogen. The pentose phosphate pathway, and in addition to this pathway, we talk about the metabolism of monosaccharides, including fructose and galactose. Okay, let's talk about digestion and absorption of the carbohydrate. So the carbohydrate enter to the mouth, and here the alpha amylase starts to digest the carbohydrate. Okay, so the carbohydrate they could be in the form of the starch or disaccharide, including lactose, sucrose, or cellulose. So some of the digestion occurs by the alpha amylase, and the alpha amylase break down the glycosidic bond, including alpha 1 to 4, meaning that it is not capable of breaking down of all the glycosidic bond. Okay, because in some carbohydrates, in addition to the alpha 1 to 4 glycosidic bond, we have other glycosidic bond as well. For instance, alpha 1 to 6, but alpha amylase is not capable of breaking down. So therefore here the digestion of the carbohydrate is not complete, and in the stomach the low pH stops the activity of alpha amylase, and most of the digestion occur in the intestine. So in the small intestine, the pancreatic alpha amylase secreted by the pancreas into the small intestine continue the digestion of carbohydrate, and at the end here in the intestine there are enzymes that are mucosal cell membrane bound, including maltase, lactase, sucrase, that are capable of breaking the disaccharide down to the monosaccharide. Okay, because the only form of carbohydrate that could be absorbed in the intestinal cells and enter to the circulation is monosaccharide. Therefore, the digestion of carbohydrate could be resulted in production of monosaccharide. Otherwise, they could not be absorbed, and under the function of the bacteria, they could be fermented to the products that leads to abdominal pain. So here in the intestine, the disaccharide under the function of enzymes mentioned here. For instance, regarding to the lactose, the enzyme known as lactase, by the aid of the water and hydrolysis, can hydrolyze lactose to galactose and glucose. It's the same for the other disaccharide here. For instance, regarding the sucrose, the enzyme is sucrase, which can digest sucrase to the fructose and glucose. Okay guys, up to here you have learned that the carbohydrates are digested in our mouth, in the stomach partially, and mainly in the intestine. As the monosaccharides are produced in the intestine, then the monosaccharides should be absorbed in the intestinal cell. Okay, so before I talk about the absorption of these monosaccharides, let's talk about the lactase deficiency. So there are persons that cannot digest lactose because they suffer from the lactase deficiency, and the lactose here in the small intestine could not be digested by lactase. Therefore, in the large intestine, the lactose under the function of bacteria could be digested, and the products that are produced here result in bloating, diarrhea, and dehydration. Okay guys, so up to here we said that the dietary carbohydrates are digested to the monosaccharide. Please imagine that here is the lumen of the intestine, okay, and this is the intestinal cell, and here we have the circulation. Okay, so for the absorption of the monosaccharide, because they cannot pass from the membrane of the cells, there are transporters. Okay, so the transporters such as SGLT, abbreviated for sodium glucose dependent transporter, and other transporters are known as GLUT. Okay, we have different GLUT. I will explain to you in the next slide. I will give you more information about the feature of these transporters. So here you see that SGLT is capable of passing glucose or galactose into the intestinal cells. So the movement of glucose or galactose via this transporter is dependent on the presence of the sodium. Okay, so this is a co-transport system, and the sodium here, the gradient of the sodium facilitate the transport of the glucose or galactose. How the gradient of the sodium occurred here? Because here there is a sodium potassium ATPase, which is not shown in the picture. So these sodium potassium ATPase, by the use of the ATP, by the use of the energy, can move 3 sodium out of the cells and enter or move 2 potassium into the cells. Therefore here we have the gradient of the sodium, and by the use of this sodium, by the help of this sodium, the glucose or galactose could be entered to the intestinal cells. Okay, so therefore SGLT, it does not use ATP directly, but it uses ATP indirectly, because regarding the gradient of the sodium, this sodium potassium ATPase has already used ATP. Therefore this is a secondary active transporter. So regarding the GLUT, so here we have the GLUT5, which transport fructose from the lumen of the intestine into the intestinal cells. So as you see, this transporter pass only fructose, and this is the facilitated diffusion, and it depends on the concentration of the fructose. As far as the fructose, the concentration of fructose is high, here the fructose could be moved into the intestinal cell. After the absorption of the glucose, galactose, and fructose into the intestinal cell, all these guys could pass through the intestinal cell to the circulation via GLUT2. Okay, so the GLUT2, based on the concentration of this monosaccharide, it can pass them to the circulation. So we talked about the GLUT as the glucose transporter, and you see here that we have different GLUTs numbered by GLUT1, GLUT2, GLUT3, GLUT4, and GLUT5. Okay, so in this table you see the tissue location of this GLUT, and there are some comments on them. So the GLUT1 and GLUT3 is present on all cells of our body, okay, and GLUT2 is present in the liver and pancreatic beta cells. GLUT4 is present in the muscle and fat cell. GLUT5 is present in the small intestine. In addition to these 5 GLUT, another GLUT exists, which is GLUT7, and it's present on the ER, or endoplasmic reticulum. When I talk about the gluconeogenesis and glycogenolysis, I will explain more on this GLUT to you guys. So the GLUT1 and GLUT3, since they are present on all cells of our body, therefore they are involved in the basal glucose uptake, and the GLUT2, which is present on the liver cells and pancreatic beta cells, it is involved in the regulation of insulin, and it removes the excess glucose from the blood. Okay, because the liver is a tissue which plays important role in the regulation of blood glucose level. Okay, because you see, then you know that the glycogen is made and is stored in the liver and in the muscle. Therefore, the muscle and the liver are the tissue that have important role in lowering blood glucose level after we had meal. And pancreatic beta cells, you know, in these cells we have insulin which is stored. Therefore, in response to the increasing of blood glucose level, the beta cell release or secrete insulin into the bloodstream in order to lower blood glucose level. That's why the GLUT2 plays role in regulation of insulin and in liver in removing the excess glucose from the blood. Okay. So how about the GLUT4? GLUT4 is present on the muscle cells and fat cells. And what is important about the GLUT4 is that the GLUT4 is insulin dependent, meaning that the GLUT4 is, let me see if I have the slide for this. Yes, here. Okay. So as you see in the title, the insulin stimulates the uptake of glucose by GLUT4. Okay. So imagine that this is the membrane of muscle cells or fat cells. Okay, so here we have a receptor, okay, and here we have the insulin. As the insulin binds to its receptor on these cells that I mentioned, so by the message which is entered to the cells, which is transferred to the cells, the GLUT4 that are on this vesicle could be transported to the plasma membrane cells. Okay, so then it enables these cells to uptake the glucose and move it into the cytoplasm of these cells. So if the insulin is not enough, or this receptor shows resistance to the insulin, so we will not have the exposure of GLUT4 to these cells. This is the problem of the diabetic patient. Okay, either the insulin is not enough, or the insulin receptor has got resistance to the insulin. Therefore, this system does not work properly. Okay, therefore, the only glut that is dependent to the insulin for the uptake of the glucose is GLUT4. Okay, how about the GLUT5? GLUT5 is involved in transporting of the fructose, as I show you in the previous slide. Regarding the GLUT, here you see that there are information about the Km. You know that Km represents the affinity of the, for example, the less Km is, the GLUT has more tendency to remove the, or to uptake the glucose. Okay, for example, regarding the GLUT1 and GLUT3, the Km is 1 millimolar, and the average level of the blood glucose is 5 millimolar. Since the Km of these two GLUT is lower to the average of the blood glucose level, therefore they are they would like to uptake the glucose even the glucose is not high in the blood. That's why these two GLUT guarantee the uptake of the glucose to all the cells of our body, although the glucose level is not very high, because the Km of this GLUT is lower compared to the average blood glucose level, which is 5 millimolar. But regarding the GLUT2, this Km is above the average blood glucose level. Okay, 3 times, 4 times higher than 5 millimolar. Therefore, these GLUT transport the glucose to the liver and pancreatic beta cells when the blood glucose is high. So, and it should be in this way, because as I told you, the liver is responsible for lowering blood glucose level, and pancreatic beta cell as well, by the secretion of the insulin. Therefore, when the blood glucose level is high, these cells should allow the transport of the glucose. And regarding the GLUT4, as you see, the Km is 5 millimolar. Here you see the schematic figure regarding the glucose transporter, the GLUT. So it is shown in green, and the glucose is shown in blue. So as the glucose is taken by this GLUT, so the conformation of this GLUT is changed a little bit, and it allows the movement of glucose to the cytoplasm of the cell. So in the first slide, I talked about the different metabolic pathway of glucose, and I would like to talk about these metabolic pathway in different cells, just because I want to make you understand that all these pathway are not present in all cells of our body. Okay, so here we have red blood cells and brain tissues. The glucose enter to the RBC via the GLUT, and it is metabolized either in the glycolysis pathway or pentose phosphate pathway. So in the glycolysis pathway, the product of the end product of the glycolysis is lactate. I mean, that's pyruvate, but then pyruvate reduced to the lactate. So how about the brain tissue cells? The glucose enter brain tissue cells via the blood, and it is metabolized in the glycolysis pathway. The product is pyruvate. Pyruvate is changed to the acetyl-CoA. Acetyl-CoA enter to Krebs cycle in order to release its energy. As you see here, the glucose 6-phosphate, the glucose in glycolysis pathway is converted to the glucose 6-phosphate, and then glucose 6-phosphate enters to the pentose phosphate pathway as well. Therefore, here we have glycolysis and pentose phosphate pathway. So you may ask me how the glucose decides to enter which pathway, because we have a variety of metabolic pathway for glucose metabolism. It all depends on the situation of the cells. Okay, I will talk about it later for you, that the pathway have the regulation. They are regulated by the hormone, by the metabolites, by the, for example, enzymes. Okay, and the cell understand which metabolic pathway should be on, which one should be off. It's all about the demand of the cell. Okay, if the cell needs the product of pentose phosphate, or the cell needs to turn on glycolysis in order to get energy. Okay, it all depends on the demand of the cell. So here you see the muscle and heart tissue cells. On right side of the slide, the adipose tissue cells. As the glucose enter to the muscle and heart tissue cells, it's either enter to the glycolysis. The product is pyruvate. Pyruvate could be converted to the acetyl-CoA, enter to the Krebs cycle, or pyruvate could be reduced to lactate. Okay, you see there are two fates for pyruvate in this cell. It depends on the presence of the oxygen. Okay, so the glucose 6-phosphate produced from the first reaction of the glycolysis here either could be entered to the pentose phosphate pathway, or it could be used for production of the glycogen. Okay, and the glycogen could be broken in order to produce glucose 6-phosphate. How about the adipose tissue cells? So the glucose enter glycolysis, produce glucose 6-phosphate. Again, it enters either to the pentose phosphate pathway to production of the glycogen, or continuous glycolysis to produce pyruvate. Pyruvate is converted to the acetyl-CoA, and here acetyl-CoA could serve as the precursor of fatty acid biosynthesis. Okay, so either it enters to the Krebs cycle to produce energy, or it could be used for production of the fat in the adipose tissue cells. So guys, here I forgot to mention that RBC lacks mitochondria, therefore the end product of the glycolysis is lactate, because the RBC lacks mitochondria. So the cells that lack mitochondria, or they have limited oxygen, they convert pyruvate to the lactate. The pyruvate as the end product of the glycolysis. Okay, so another cell is the hepatocyte, or liver parenchymal cells. So as you see, we have all the metabolic pathway related to the glucose on in the hepatocytes. The glucose enter to the hepatocyte. Here we have the glycolysis. The pyruvate is formed. Either produce lactate or produce acetyl-CoA. What happens to this acetyl-CoA? Either it enters to the Krebs cycle, or it is used to produce fat, and the fat is packaged in a lipoprotein known as VLDL, and VLDL is exported out to the circulation. Okay, so here you will learn in the session of the lipid metabolism. Okay, so here you see that there is an arrow from pyruvate to glucose. This is the gluconeogenesis pathway, the pathway which converts non-carbohydrate substrate to the glucose. And here you see that the glucose 6-phosphate enters pentose phosphate pathway or oronic acid pathway. So guys, you see that oronic acid pathway takes place only in the hepatocyte. It produces glucuronate. So the glucose 6-phosphate could be converted to the glycogen, the polymer of the glucose, the storage form of the glucose in the liver, and the glycogen could be broken down to produce glucose for the hepatocytes.